Hello, everybody. Welcome to Ms. Maguire online video lecture. And today we are covering human genetics, gene action from DNA to proteins. This video lecture will be divided into two parts. In the first part, we will talk about protein synthesis. And in the second part, we will talk about folding of proteins and how misfolded proteins can cause diseases. Right, so let's go ahead and begin. So objectives for this chapter. After studying this chapter, you will be able to explain the relationship between proteins and amino acids, explain the relationship between genes and proteins, list the major types of RNA molecules and their functions, explain the importance of transcription factors, list the steps of transcription, discuss how researchers deduce the genetic code, list the steps of protein synthesis, define the four components of a protein's shape, and explain the importance of protein folding. Now, proteins are polymers that are made of monomers, or what we call building blocks, called amino acids. So proteins are made of amino acid. Genes control the linking of amino acids into proteins. 20 amino acids are required for the body to produce its own protein molecules. 9 of the 20 amino acids considered to be essential. That means you cannot synthesize them. You need to get this 20 amino acid with your food. Each amino acid has a central carbon that bonds to amino group and acidic group. That's why we call it amino acids, because we have NH2 group, that is amino group, and COOH group, that is acidic group. It's also attached, the central carbon also attached to a hydrogen atom and variable R group. It is the R group that distinguishes 20 different amino acids. So let's go ahead and look at structure of protein. So over here you can see a central carbon, and it's attached to hydrogen, amino group, and acid group. So this part of amino acids is the same for 20 amino acids. So all 20 amino acids will have the same central carbon, amino group, acid group, and hydrogen. This is our group that will be different for each amino acid. So this is arginine. So you can see the structure of our group. (20,325) or so proteins that the human body has are coded by protein coding genes, termed the exome. Now, in your DNA, you have special genes, right? You have a gene that code for that many proteins, and this part of the DNA we call exome. Proteins serve many vital functions. Some of these functions are contractile, regulatory, enzymatic, structural, transport, immunity, and clothing. Proteins are composed of one or more long chains of amino acids called polypeptides. A protein's three-dimensional shape is called conformation. So proteins must have a very precise three-dimensional shape, or they must have precise conformation to function properly. Cell uses two processes to manufacture proteins using genetic instructions: transcription, synthesis an RNA molecule, and translation, that uses the information in this RNA molecule to manufacture a protein by aligning and joining specific amino acid. Folding of the proteins into specific 3D form necessary for its function. Now, this is the diagrams that represent eukaryotic cell. Eukaryotic cell means you have a nucleus inside a cell. Now, DNA located inside the nucleus. Now, DNA need to be transcribed, so you need to make a copy of a gene. Not entire DNA will be transcribed into RNA, but a part of DNA that code for specific protein, a gene, we will transcribe into RNA. This RNA will leave the nucleus through nuclear pores, and inside the cytoplasm, information now from this RNA molecule will be used to synthesize proteins, and this is called central dogma. Central dogma is a directional flow of genetic information from DNA to RNA to proteins. And another process shown over here is the replication of DNA. When the cells make copies of DNA molecule, and they need to replicate their DNA before they divide. So they can pass one copy of DNA to one daughter cell and another copy of DNA to another daughter cell. Now, when we're talking about replication, we replicate entire DNA. When we're talking about transcription, we make a copy of only a part of DNA, that specific gene that we need to make specific protein. RNA is a bridge between gene and protein. Bases of an RNA sequence are complementary to those of one strand of double helix that's called template strand. So remember, DNA is double helix, so it's I don't want it to disappear. So here's the DNA molecule that made of two strands, and let's say one strand we will use to make RNA. So this will be used to make RNA. So that means this strand over here is called template. Then non-template strand, now this strand will be called coding strand. So that's a coding strand right here, and this one is template because we will use this one to make RNA molecule. Now we need enzyme to make RNA from DNA, and this enzyme is called RNA polymerase that will build this RNA molecule. Okay, so that's what we just discussed. You can see on the diagram here. So we inside the nucleus and we're talking about transcription. So here's our DNA shown here in blue. So it's a double helix. Here's our DNA template strand, and DNA coding strand. Now DNA template strand, the same as the coding, have the bases, right? Remember, DNA has A, C, T, and G. So here's your A, C, A, G, T, A, G, C, C. Now those will be complementary. A always pairs with T, C with G, A with T, G with C, right? But the DNA will be unwind, so it will be open, and RNA will be synthesized using DNA template strand, and it will be synthesized using this rule of complementarity of nucleotides. So if you have A, when we make RNA, we're gonna use U because RNA doesn't have thymine. So instead of T, we will use U. So if the template is C, then RNA nucleotide will be G, right? C pairs with G, G pairs with C, A pairs with U and T pairs with A. So this messenger RNA is a transcript of DNA molecule. So that's specific and not entire DNA, but I already told you it's a transcript of a specific gene. There are two types of nucleic acids, DNA and RNA. Both consist of sequences of nitrogen-containing bases joined by sugar phosphate backbones. However, they differ in several aspects. So DNA is usually double-stranded, RNA usually single-stranded. DNA use T or thymine as a base. RNA doesn't have T, it has uracil as a base. DNA has deoxyribose sugar. This is where the name came from, deoxyribonucleic acid. RNA uses ribose. It's a ribonucleic acid. DNA maintains protein encoding information. RNA carries protein encoding information from RNA and controls how information is used. DNA cannot function as enzyme. RNA can function as enzyme. DNA persists. RNA is short-lived. So DNA stores RNA and protein encoding information. So DNA has information to make RNA and to make proteins, right? And transfer information to daughter cells. So here's the DNA, it's double helix. Sugar of the DNA is deoxyribose, and bases that are used, T, C, A, and G. Right, so here's a show you example of nitrogenous base. When RNA is generally single-stranded, different sugar ribose, it's similar. So if you look over here, deoxy means one oxygen less. So you don't have this oxygen, and now instead of ribose, you have deoxyribose. And it uses uracil instead of thymine, but cytosine, adenine, and guanine are the same in DNA and RNA. And you can also see the difference between thymine and uracil, right? So that's a part that is different. RNA carries protein encoding information and helps to make proteins. We have different types of RNA molecules. mRNA or messenger RNA encodes amino acid sequences. So messenger RNA have a code how to make proteins, how to put this amino acid in a precise sequence. So it's about 500 to 4,500 nucleotides. It's made of this many nucleotides. rRNA or ribosomal RNA is a smaller 100 to 3,000 nucleotides associated with proteins to form ribosomes with a special protein. So it attaches itself to special proteins called ribosomal subunits and form ribosomes, which structurally support and catalyze protein synthesis. So this supports and catalyzes protein synthesis. tRNA or transfer RNA, 75 to 80 nucleotides, transport specific amino acid to a ribosome for protein synthesis. So here we already mentioned that we have different types of RNA. One of them is mRNA that carries information that specifies a specific protein. 3 mRNA bases in a row form a codon which specifies a particular amino acid. So we're gonna look on the next slide what this codon means. Most mRNA are 500 to 45,000 bases long. Differentiated cells produce certain mRNA molecules called transcripts. Information in these transcripts is used to manufacture and encode proteins. So let me Just a second, see if we have this messenger RNA and codon. We do, but maybe I'm gonna draw it for us so we can see it better and understand better. So here's the molecule of mRNA. So that's mRNA. And you know, as a nucleic acid, it's made of nucleotides. So here's gonna be my nucleotides. And we have A, C, G, U, and maybe again U, and again A, and again A, and again C, and G, and so on, right? So mRNA made of nucleotides A, U, C, and G. Now, if you divide this mRNA into into parts that includes 3 nucleotides. Look, this one includes A, C, and G. This is called codon. So that's codon. And then another 3, UUA. There's gonna be another codon. UUA. So that's another codon. This is another codon, ACG. ACG. Right. And so on. So these 3 mRNA bases, 3 bases in a row called a codon, and each codon codes for specific amino acid. Then we have our rRNA, that is ribosomal RNA. Okay, I'm trying now to get rid of this red pen. So most ribosomal RNA are 100 to 3,000 nucleotides long, associate with certain protein to form ribosomes. So we already said it attaches it to certain protein to form ribosomes. Ribosomes consist of 2 subunits that join during protein synthesis. rRNA provide more than structural support. Some are catalysts, and other help align the ribosome to mRNA. So here's the structure of actually a ribosome. So that's a ribosome that is made of proteins, large subunit and small subunit. Here we have about 47 proteins in a large subunit. We have 32 proteins in a small subunit. And ribosomal RNA, so that it will be attached to mRNA. So that's gonna be, let me get, so that's gonna be our M messenger RNA that ribosomes will attach to messenger RNA. But the subunit ourselves going to be associated with rRNA. So then we have tRNA that binds an mRNA codon at specific amino and a specific amino acid. tRNA are the smallest RNA, only 75 to 80 nucleotides long, and the 2D shape is a cloverleaf shape, and the 3D shape is an inverted L shape. So I'll show you on the next next slide, the structure of tRNA. Now, tRNA has 2 ends. One is anticodon that is complementary to messenger RNA, and opposite and strongly bonds to a specific amino acid. So here's the structure of tRNA. So we can see we have a loop. So it does look like a clever. And on one side, we have 3 nucleotides, AAG in this example. This is called anticodon. And anticodon of the tRNA. So that's just, this is tRNA. So anticodon of tRNA gonna bind to messenger RNA. And on another side, we will have amino acid accepting end. So amino acid gonna bind to this end. And it'll make sense when we start talking about protein synthesis. So enzymes attach amino acids to tRNAs that bear the appropriate anticodon. So here's our messenger RNA. So remember, all the RNA, the same as DNA, made of nucleotides, and each nucleotide has a nitrogenous base. So here's our base A, U, and G. That's on a messenger RNA. Now tRNA must attach itself to mRNA. So how tRNA attaches itself to mRNA? Well, because the tRNA has anticodon. And anticodon is complementary to a codon of mRNA. You see, A always binds to U, U to A, and G to C. So this tRNA is like this tRNA is complementary to this codon. Codon and anticodon, they match. And this UAC, if tRNA has codon UAC, it's always gonna carry this specific amino acid, right? Methionine. Now this is different tRNA. It has different anticodon, so it can bind itself to complementary codon, right? And complementary codon is GGA because G pairs with C and A pairs with U, and it carries a different amino acid. Now here, anticodon is ACA, so it's gonna bind to UGC because A always binds with U, G with C, and U with A. And it carries a different amino acid. Now this tRNA cannot bind to this codon, right? It cannot bind it because G will never pairs with A. So G will pairs with C over here, but A will not pairs with A, right? So the point is that tRNA can only bind to complementary codon on mRNA. Now we also need transcription factors to start transcription of DNA. A transcription factor interact and form apparatus that binds DNA at a certain sequences. They initiate transcription at specific sites of chromosomes. They respond to signals from outside the cell, and mutations in transcription factors may cause a wide range of effects. Now, the transcription of DNA, and I just want to remind you what transcription is. So you start with DNA. DNA has the information how to synthesize proteins, but DNA is a large molecule and it's located inside the nucleus. It has information how to make 20,000 of different proteins, and you need maybe only one protein. Maybe you need right now growth hormone or some insulin. And so you need just very specific protein at this moment. So you cannot use entire DNA that codes for thousands of proteins. So that's why your cells will only look at a specific part of DNA that we call gene, specific part of DNA, and they're gonna make a copy of it, copy of this gene, and now this copy cannot be a gene, right? We just make a copy of it. This is gonna be messenger RNA. And this process of making messenger RNA from a gene of DNA is called transcription. So we're gonna rewrite, we're gonna transcribe information from a gene into messenger RNA. Now the transcription itself is made of 3 steps. It's made of initiation, elongation, and termination. So you're gonna initiate transcription, you're gonna make messenger RNA longer, that's what elongation, and then you're gonna stop. In transcription initiation, transcription factors and RNA polymerase are attracted to a special part of DNA called promoter. So let's look where this promoter is located. Okay, so we look over here and then we get back. So here's our, so here's the DNA molecule, right? This is just the template strand. So it's just one strand. And at the specific part of a DNA, we will have a sequence that is GGTATACCC, right? Especially this part over here is called Tata box. So that's a sequence that is promoter. Now we will have binding protein, transcription factor, and RNA polymerase binds to this promoter. And after that, we start transcription. That makes sense. Now, we need these binding proteins and transcription factors to start the process, and RNA polymerase gonna make our messenger RNA. Right, so now let's go back, and that's what I said, that in transcription, that's going to be initiation. In initiation, transcription factors, right? So this one, transcription factors, binding proteins, and RNA polymerase attaches to promoter. They attracted to promoter. Then we have elongation. In elongation, enzymes unwind the DNA double helix. Free RNA nucleotides bond with exposed complementary bases on a DNA template strand. RNA polymerase add the RNA nucleotides in the sequence of DNA specifies, and terminator sequence in the DNA indicates where the gene's RNA encoding region ends. Right, so we look at the picture and everything will make sense. So what is our goal? Our goal is here to, again, if you have DNA that is made of 2 strands, we have a template strand, and we just take a part of it, right? This part, for example, and this part code for protein. So that's our DNA, and our goal is to make protein. Now, to make a protein, we need to link amino acids together, right? So that's our amino acid. We're going to link them together to make protein. Now, what amino acids need to be attached? Because we have 20 different amino acids. So we don't know which one we need to attach in what order. We need instruction. So instruction is right here in this piece of DNA that we call gene. It codes for this particular protein. So first, we need to make a copy of this gene. So we need to make a copy of this gene called messenger RNA, right? So here's our DNA molecule. And on this DNA molecule, in a specific part, we will have this Tata box. And this area is called promoter. So we initiate the process. We initiate the transcription. We attach binding protein, transcription factors, and RNA polymerase. So that's our initiation. We didn't synthesize mRNA yet. We just initiated the process, right? So after that, we have elongation. So in this example, so we already passed the promoter, right? We already passed the promoter. So here's our RNA polymerase. So it starts moving along the DNA. It's open up. See how it's open? It's open up the helix, and it's gonna bind these nucleotides, attach them to each other, but not just any nucleotide, but the one that is complementary to the nucleotide on a DNA strand. So for example, let's say this was A. Okay, for example, this was A, right? Nucleotide, this was T, this was C, this was C again, this was G, and this was A again, right? Then RNA polymerase gonna put here, see this green part? It's not gonna put any C or G, because what complementary with A? What complementary with A is U. Well, remember A with T and C with G, but because RNA doesn't have T, instead of T, we're always gonna put U. So over here, we will put U. T, you look over here, right? Well, T complementary with A, so we're gonna put A. C, we're gonna put G, we're gonna put G again, and we're gonna put C, and A again, U. Right, so here's our mRNA. This is our sequence somewhere we made A, or U, A, G, G, C, and U, right? So, so here's our elongation. See how mRNA became longer and longer until RNA polymerase reaches this part of DNA that's called terminator. When it reaches the terminator, everything splits apart, right? RNA polymerase will move away, messenger RNA will move away, and the DNA will close up, right? And we reach this special sequence terminator. That means we made messenger RNA, right? Okay, so this is the process of transcription that is made of initiation, elongation, and termination. Now, several messengers RNA may be transcribed from the same template at the same time. So here's our DNA template strand, and we have several RNA polymerases moving along and making a messenger RNAs. Now, why do we need this messenger RNA? Because we want to carry this message that was coded inside the DNA. We can carry this message to the ribosomes, and we make proteins based on this information inside this messenger DNA, inside the sequence of the nucleotides of messenger RNA. In eukaryotes, messenger RNA must exit the nucleus to enter the cytoplasm, right? So messenger RNA is made inside the nucleus. Several steps process free messenger RNA into mature mRNA. So what we just did, it was actually pre-messenger RNA. We need to transform it into mature mRNA. So we're gonna add a cap. It's called a 5' cap, methylated cap. So it will be the sequence of nucleotides. And we need this cap as a recognition site for protein synthesis. And on a 3' end, we're gonna add a poly A tail, necessary for protein synthesis to begin and stabilize the messenger RNA. So if we look over here, right there, and I will go back. So here's our DNA template. Just ignore this exon and intron for a second. But here's our DNA template. We have a transcription, so we made a copy of it. And then look, we added this 5' cap and 3 poly tail. And the poly A tail is just adenine, right? It's just nucleotide with adenine. So that's when we make it mature. Right? But something else needs to happen. We need to splice the messenger RNA, because when we made this mRNA, it has introns that do not code for proteins and exons that that code for protein sequences, for amino acid sequences. Sorry. Right, so we need to cut out those introns and only leave exons as a part of messenger RNA. Note that introns may outnumber and outsize exons. So messenger RNA then is proofread and the mature messenger RNA is sent out of the nucleus. Introns and exon organization of genes maximize genetic information. Different combination of exons of about 95% of human genes encode different version of a protein termed isoforms. The mechanism of combining exons of a gene in different way is called alternative splicing. Cell types can use a version of the same protein in slightly different ways in different tissues. And so, so here's the entire process that we just described. So we started with DNA, and we need to make mature mRNA, and this mature mRNA need to leave the nucleus. This shown in blue is nucleus, and this is cytoplasm. So DNA has two strands. One of them is template strand. Through transcription, we make a copy of this strand. So you see exon A, intron A, intron A, exon, intron 2, exon C, right? So we made this pre-messenger RNA. Then we're gonna modify it. We're gonna add our 5' cap and poly A tail, and then we're gonna splice it. So we're gonna take this intron out, and we're gonna join exons together, because introns, they do not code for protein. So those information we don't really need. We just need the information of exons. So when we, after splicing, we get our mature mRNA with a cap and a tail, and it will be transported out of nucleus into cytoplasm. Now for second part of protein synthesis that's called translation. Now, what is translation? So we just described transcription. Transcription is the process of making transcript in the form of messenger RNA from DNA. So we take a gene or part of DNA and we make a copy of it. We call it messenger RNA. That's transcription. Now, when we synthesize messenger RNA, we modify it and we splice it. We added cap and tail. We cut out introns. Now this messenger RNA is ready. It's inside cytoplasm, and it's ready to for protein synthesis. And process of linking amino acids together. Oh, here's our amino acid: lysine, serine, valine. When we link them together, this process is called translation. Well, not when we link, but when we translate information from messenger RNA into specific amino acids. That's what translation is. Translation assembles a protein using the information in the messenger RNA sequence. Particular messenger RNA codons correspond to a particular amino acid, and process occurs on the ribosome. So here's our, again, that's our goal. That's our central dogma. We go from DNA, we have information how to make insulin here, for example, in the DNA. We need to find this gene, make a copy of this gene through transcription, and a copy of this gene is called mRNA. Now, we use this now information in mRNA to translate it into protein. So here's our DNA template. And for example, here's our gene, right? So this is our gene, TTCAG, TCAG. That's a gene. We make messenger RNA. We use RNA polymerase. How do we make it? Well, we cannot just put anything here. For example, we cannot put, I don't know, C here, right? Because it's not complementary. We will make sure that we put a precise nucleotide. T pairs with A. So here we're going to put A, G, U, C, A, G, U, and C, because they're complementary. T pairs with E, T with E, C with G, A with U, G with C, T with A, and so on. Right, so that's our messenger RNA. Now remember, if we divide messenger RNA into nucleotide, into 3 nucleotide sequences, that's called codon. Each codon codes for specific amino acid. And that's our translation. When we translate information from messenger RNA into proteins. The correspondence between the chemical language of mRNA and proteins is called genetic code. In 1960s, researchers uses logic and clever experiments on simple genetic system to decipher the genetic code or discover this genetic code. So now let's look what genetic code is. So today, scientists know that if you have, so if you have your messenger RNA, right? So here's your messenger RNA. So it's mRNA. And they know if, for example, you have U, C, and U, this code, right? This is a codon, UCU. They know that UCU gonna code for amino acid serine, right? If you have, for example, GUU, right here, that's code for valine. So each amino acid, and we have 20 amino acid. So those amino acids are coded by a different triplet. So that's called genetic code, right? So here's our first letter, here's our second letter, and this is the third letter. So if you see that UC code for phenylalanine, UUA and UUG for leucine, UCU, UCC, UCA, UCG, all 4 code for serine. This all 4 codes for proline, and so on, right? So we know for all these 20 amino acids, and if you count over here, it's gonna be a 20 amino acids, right? That we have our 3 nucleotides that is called codon that specifically code for this amino acid. Now, if you look at some, this one, AUG, this is our methionine that is a start codon. So to start making any protein, you need to start with AUG. And if you look over here, some codons, UAA, UAG, or UGA, for example, they do not have any amino acid. They mean stop. So when we reach this codon, that means we done this protein polypeptide synthesis. Okay. So the characteristic of genetic code, it's a triplet code. 3 successive mRNA bases form a codon. We have 64 codons. Altering the DNA sequence by 1 or 2 bases produces a different amino acid sequence due to disruption of reading frame, and it's termed frame shift mutation. So let's see exactly what it means. So, for example, this is our original RNA sequence. Right, so we have a DNA. How we make this RNA? Because we made this copy from DNA. So here's our RNA sequence at GAC, 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 and so on, right? So they give us the same codon. So all code for aspartic acid. So the same amino acid. Now let's see what happens if we insert a different. So if we insert one extra nucleotide over here, because we read it by letters of 3. Now, if we insert one extra, then we're gonna alter amino acid sequences after this insertion. If we insert 2 bases, we again gonna alter the sequences. But if we insert 3, then the sequence was altered. We have only one extra amino acid, but then it's restored. Right, so you can see how it works. Now, let me give you an analogy. For example, you have this sentence. The cat ate the rat. Right, so you see we have 1, 2, 3, 4, 5 words. Each word has 3 letters, and each word codes some information. So when you read it, you understand the cat ate the rat, right? That tells you something happened, right? So that imagine that in our RNA, we have words, and each word is made of 3 letters. We cannot have words in RNA that made of 4 letters or 5 letters or 2 letters. All these words, right, and words in parentheses in RNA made of 3 letters. Now, and the ribosome gonna read it, and ribosome can only read by 3 letters. Now, let's see what happens if we insert one letter. For example, we insert letter T over here. Just insert letter T. But now what's gonna happen when our ribosomes read this, it's gonna read by 3 letters. So the first gonna be Z, and then it reads 3 next letters. So it's T, C, A, then 3 letters, T, A, T, then E, T, H, and then E, R, E, R, A, and then we have T. So now we have like sentence that doesn't make sense. Z, T, Tika, Ted, is, era. So it's no ribosomes. When ribosomes read this, there is no way we carry the same information. And this is, by the way, called mutation. It's a frame shift mutation because we shifted the reading frame. And now if ribosome gonna make protein using the wrong sequence and wrong template reading frame, the protein is not built correctly. That means proteins will not function, and as a result, somebody can have a disease or even die. So this is what it's called when we alter the DNA sequences, and that's what we call mutation. When we alter sequences of DNA, when mutation happens, then we can produce a different reading frame that is called frame shift mutation. And this mutation will result in the synthesis of the defective proteins. Okay. Another, so that's our characteristic of genetic code over here. It's a triplet code that we just described. Another characteristic of genetic code is not, it's non-overlapping. It means the same letter is not used for 2 different codons. So that means if we have, for example, A, U, C, G, and then we have A, U again, and so on. So our codon gonna be AUC, right? And we cannot use this C to make another codon, CGA. We will not have codon. So we have A, U, C, and the next codon cannot be CGA. So the same letter cannot be used in a two different codons. So next codon is gonna be G, A, and U. That's what means it's non-overlapping. And it includes controls, includes directions for starting and stopping translation. Also, genetic code is universal. That means that your genetic code as a Homo sapiens is the same as a genetic code in a very simple bacteria, or in a plant, or in other animals, or in a protist, or in a fungus. So all living organisms use the same genetic code. It's evidence that all life evolved from a common ancestor, and different codons can specify either the same amino acid, then they called synonymous codons, or non-synonymous codons encode different amino acid. What it means, if you look over here, that we have this UUU, UUC, UUA, and UUG, or I'm sorry, there's two different amino acids. Look over here, CUU, CUC, CUA, CUG, that all code for the same amino acid, leucine, so those are synonymous codons. And CUG and CCG are not synonymous because they code for the non-synonymous because they code for different amino acid. Okay, so so that we describe the transcription, right? So everything we talk about, let's go back, that was a transcription of when we make, this is my, I'm looking for my cell here. Right, so here's the cell, and inside the nucleus, we go from DNA to RNA, and it's called transcription. So we made this mRNA, and now we know this mRNA have codons, and those codons, 3 letters of nucleotides, code information for a specific amino acid. So the next, we need to move to translation. Now, transcription is when we go from DNA to RNA, and we call it transcription because we don't change language. We go from nucleotides, DNA is made of nucleotides, and RNA is made of nucleotides. So we just transcribe it. So we rewrite it very much. But when we are talking about translation, we go from nucleotides, right? So nucleotides and mRNA to amino acids. So it's like we're changing the language. And when you change the language, you need to translate, right? From one language to another. That's what translation is. So a messenger RNA leaves the nucleus, moves to the cytoplasm, and this is where translation begins. Now, translation also has initiation, elongation, and termination. Right, so let's look at this initiation of translation. The leader sequence of the mRNA forms hydrogen bonds with a small ribosomal subunit. So messenger RNA gonna form a bond with ribosome, but first with a small ribosomal subunit. Then the start codon, that is AUG, attracts the initiator tRNA that carries methionine. And this completes initiation complex. So here's what happened. So we have our messenger RNA, right? Now this messenger RNA so the beginning of messenger RNA will attract a protein that is called small ribosomal subunit. So you see how it's gonna attach itself to mRNA, and we have a first tRNA. First tRNA is always UAC because it's gonna bind to AUG, right? So that's our first amino acid, always methionine. So here we have a small ribosomal subunit, we have messenger RNA, and we have this initiator tRNA that carries methionine. And this is our initiation complex. So we're gonna start translation. After that, a large ribosomal subunit binds to this initiation complex, and TRNA bearing a second amino acid, in this example glycine, forms hydrogen bonds between its anticodon and the messenger RNA's second codon at the site A. The first amino acid, methionine, occupies the P site. So let's see on a diagram here. Now, let me just draw instead of. So here's our messenger RNA. Here's our small ribosomal subunit and large ribosomal subunit. We already, in initiation, we had small subunit mRNA and the first amino acid that is methionine. So right here. Now, where methionine is attached is called P site. And then we have another site called A site. Right, so when we form this initiation complex, we have another tRNA that's gonna bring another amino acid. In this example, tRNA has anticodon CCU, see over here, C and U, and CCU carries glycine. So now, so this tRNA kind of, it's tried to attach here, it can't, right? It tried to attach itself here, can't, right? So it attaches itself at a specific area where we have match between codon, and this is our codon, and anticodon. And this is our site A, right? So in this situation, it's a glycine. So we have methionine and glycine. Now what's gonna happen, this ribosome is actually gonna move. So you see how this glycine was in the site A, now it's in the site B, because the ribosome just moved. And glycine, this tRNA carrying glycine is just shifted from A to P site, right? Now the methionine brought by the first tRNA forms a peptide bond with the glycine. And then tRNA just moves away. So here's our tRNA. It's donated this methionine. It attaches methionine to glycine, and it just moved away. So it's going to pick up another methionine for another protein synthesis. Then, right, then we have our A site vacant, because our ribosome moved. Now look, there is nothing here. Now we can attach another tRNA. Now what tRNA you can bring over here? Well, we look at this codon, U, G, and U. So the only tRNA that's gonna fit in this A site is the one with anticodon ACA, because U pairs with A, G with C, and U with A. And UCA carries another amino acid that is cysteine, right? So, so what it says, then the second tRNA, so the, the third tRNA arrives in this example carrying the amino acid cysteine, at a temporary vacant A site. Then a fourth and then a fifth amino acid linked to the growing polypeptide chain. The process continues until reaching a stop codon. So you can see we have more and more tRNA bringing a specific amino acid. The chemical bonds form peptide bonds formed between the amino acid, and we making the chain of amino acid that is a polypeptide, right? And the polypeptides then became proteins when they fully folded into precise three-dimensional shape, right? Okay, so that's our translation. Now then, we need to terminate the translation. It occurs when a stop codon enters the 8th site on the ribosomes. A protein release factor attaches to a stop codon. So over here, we don't have another tRNA. We have this protein called release factor attaches to a stop codon. The ribosomal subunit separates, so you see how they separated, and recycled, a new polypeptide over here is released. Right, so we pretty much made a polypeptide. Now, multiple copies of a protein can be made. All right, so you see over here, so that's our messenger RNA, and we have several ribosomes reading this mRNA and using information in mRNA to synthesize the proteins. Okay, then when we made this sequence of amino acids, we make a polypeptide. To make a functional protein must fall into one or more 3D shapes or conformations based on attraction and repulsion between atoms of proteins and interaction of proteins with chemicals in the environment. So we have 4 levels of protein structure: primary, secondary, tertiary, and quaternary. So here's our primaries. So the primary structure is just the sequence of amino acids. So it's shown over here. It's just the sequence of amino acids. That's our primary structure. Then secondary structure: loops, coils, sheets, or other shapes formed by hydrogen bonds between a neighboring carboxyl and amino groups. So here, this is amino group, this is carboxyl group, and those dots represent very weak chemical bonds called hydrogen bonds. So we either form the alpha helix or beta pleated sheets. So we start folding a protein. Then the tertiary structure is a 3-dimensional form shaped by bonds between R groups. So remember, our amino acids, they all have different R groups. Now those R groups start interaction and between R groups and water molecule, and we have a tertiary structure. See, that's a helix, that's a beta pleated sheet. Helix, beta pleated sheet. Helix again, beta pleated sheet. So we combine these secondary structures. We combine these loops and coils and sheets together in one functional 3-dimensional proteins that has tertiary structure. Now, some proteins only have tertiary structure and they're already functioning proteins. The bigger proteins, they need a quaternary structure. This is a protein complex that form between separate polypeptides. So we'll take one of these. So here's the one, then we have another. So we have 1, 2, 3, 4 polypeptides over here that form functional protein with quaternary structure. Right, so primary structure is a sequence of amino acids in a polypeptide chain. Secondary structure: loops, coils, sheets, or other shapes formed by hydrogen bonds between neighboring carboxyl and amino groups of amino acids. Tertiary structure: 3-dimensional forms shaped by bonds between R groups, interaction between R groups and water, and quaternary structure: protein complex formed by bonds between separate polypeptides. Now, the proteins must be folded precisely in this 3-dimensional shape and form. If we have misfolded proteins, then the protein might not function properly. Many misfolded proteins causes diseases and even death of an organism, right? That's why the shape of protein is extremely important. Now, what can happen if a protein is not folded correctly or protein misfolding? We will discuss in the second part of this lecture. Right, so now thank you for watching, and I hope it was helpful.